Good morning, girls. Welcome back to web-based learning platform SoElearn. Today we are going to study with another type study in vascular cryptogams is Marselia, which is also known as the clover fern. If you see the classification of Marselia, it belongs to Sphelophyta, Pterophyta, classes is Leptosporangiopsida, order is Marsiliales, family is Marsiliaceae, and the genus is Marselia. Regarding the morphology of the sporophyte, Marselia, which is known as the clover fern, it is creeping in the mud and poses root, rhizome, and leaves. Rhizome is horizontal, and the leaves horizontal structure with fairly long internodes. From the rhizome, leaves and roots are given out of the nodes. The roots are adventitious, given out from the nodes, and from the underside of the rhizome, they absorb minerals, salt, and water, anchor sporophyte in the mud. The leaves are alternately produced from the nodes on the rhizome and are arranged in a distichous manner. Each mature leaf show long petiole with four leaflets at the tip. The leaflets are actually a pair is placed a little higher than the other in an opposite decussate manner. Each leaflet show very minute petiole which is subsessile, and the leaflet is obovate in shape and the entire or serrate margin, round apex and dichotomous venation. This is the structure of morphology of the sporophyte, having showing the but the stem, the roots, and the leaf, where the stem is the rhizome, which is horizontal. From it, the nodal regions arises the leaves, which have a long petiole and what you call the four leaflets are present. The leaflets are unequal in size, pair, which are decussate, and beneath are present the root at the from the nodes beneath from the rhizomes arises for the roots. So regarding the internal morphology, if or the anatomy of the root, it is differentiated into epidermis, cortex, and the stele. The epidermis is single layered and with a syncuticle, and beneath the epidermis is present the cortex, which is differentiated into outer parenchyma, inner outer outer cortex, which is what you call the made up of parenchyma. Inner to parenchyma is present the middle cortex. This inner cortex middle cortex is made up of parenchyma cells, and inner to the parent is the innermost cortical region is made up of the sclerenchyma without any intercellular spaces. Inner is the endodermis, which is single layered and pericycle, and there the radial vascular bundles are present, which are diarch in condition. And this is a protostele. The metaxylem is in the center, and petroxylem is towards the periphery. Regarding the anatomy of the rhizome, if you see the transverse section, it is circular and it consists of outer epidermis. Center in the center is the cortical region, where the cortex is having the outer outer parenchyma, which we call it as a hypodermis. Beneath is present the middle cortex. This middle cortex is made up of parenchyma and parenchyma, parenchymatous region, and inner parenchymatous, also made up of the parenchyma cells, and in between the two parenchymas is present the sclerenchymatous region. Then the inner is the endodermis. The stele is amphiploic siphonostele, where amphiploic siphonostele means the xylem is surrounded by the phloem. So here when you see the xylem, you can see there is a xylemin and the phloem on the outer and the inner side. The endodermis is also found in this. So amphiploic means the phloem towards the outer and inner side. Similarly, the endodermis is also found inner and outer side, and the pericycle is also found. So such kind of stele we call it as the amphiploic siphonostele, and the pith is present in the center and can be parenchymatous in case of the aquatic plants or sclerenchymatous in case of the terrestrial plants. If you see the TS of the petiole, it is again differentiated into epidermis, cortex, and the stele. Epidermis is single layered without any intercellular spaces. Then inner to the epidermis is the cortex, where it is made up of outer parenchymatous cells, middle erenchymatous cells, where the erenchyma is again septate, and the inner cortex is made up of parenchyma. The inner to the is present the endodermis, which is the limiting membrane of the cortex, and then starts the pericycle, which is single layered, and then the stele is protostele, which is plectostele. Means xylem consists of two plates, and to which metaxylem is in the center, and the protoxylem towards the periphery, and the phloem is surrounded to the xylem. If you see the internal morphology of the sporophyte of the leaflets have upper and lower epidermis, and in between the mesophyll cells are present, where the mesophyll cells are differentiated into upper palisade cell and dorsiventral erenchyma in the lower cells. So vascular bundles are concentric and are arranged in parallel series and in the lower then in the lepers. So this is the structure where you can see the isobilateral structure of the mesophyll cells. Regarding the reproduction is of three types. One is vegetative, which takes place by the tubers, which are arranged with the node regions of the rhizomes, where the some of the nodes will just fuse it up and they form the stored food, and they act as the tubers. Then comes the asexual reproduction. Asexual reproduction takes place by spores, where the spores are heterosporous and they are called the small, but the spores which are small in size are known as the microspores, and the spores which are large we call as the megaspores. So these microspores are found in the Microsporangia, and the megaspores are found in the Megasporangia. These sporocarps are green in color, where they are found near the on the rhizomes on the nodal region. The sporocarp consists of the stalk and the tubercle and a bulbil-like structure called the sporocarp. So if the young sporocarps, if you see when young, they're soft and green, and the mature one are dark brown or hard, and they can withstand the desiccation up to 20 to 25 years. They have a pedicel or stalk and a body and a point of attachment called the raphe. The distal end of the raphe, they have a teeth-like projections known as horns. The sporocarp wall is hard, thick, resistant, differentiated into three layers: outer epidermis with sunken stomata, middle thick wall palisade, and inner thin wall palisade. If you see the vascular supply of the sporocarp, the vascular supply, when you see, it has a dorsal bundle which enters the sporocarp near the lower horn and constitute forward along the upper side, forming the midrib, which are known as the dorsal bundle. From the midrib, the lateral branches arises, which are called as the lateral bundles, which pass on to both sides of the sporocarp, and then the placental bundles develop from the point of forking of the lateral bundle, which enter into the receptacle bearing the sporangia and dichotomizes. The sporocarp are bivalve structures with close network of vascular system. Fibrous sporophyll with marginal sori developed from two folded pinnae. The sori are graded type. Megasporangia in upper rows are found in the upper rows and the microsporangia in the lower rows, and the development is leptosporangiate type of the sporangial development. Leptosporangiate is a condition where group of cells will divide and form the sori or the sporocarp. Regarding the internal organization or the anatomical structure of the sporocarp is taken as vertical longitudinal section or the horizontal longitudinal section or the vertical transverse section. So in the VLS, section is cut vertically, but the sporocarp is cut longitudinally. While in horizontal longitudinal section is cut horizontally, but the sporocarp is cut longitudinally. And in vertical transverse section is cut vertically, but the sporocarp is cut transversely. Regarding the vertical longitudinal section, there are many seri arranged in vertical rows. The either Megasporangia or Microsporangia are visible. Each sorus is surrounded by an indusium. The development of sori is of gradated type. The gelatinous mucilage ring is more prominent in the dorsal side. In the HLS section, each sorus is cut transversely. The sorus is elongated structure covered by delicate indusium. Sori are graded type with basipetal manner. Sorus consists of rows of Megasporangia and Microsporangia on either side, and mucilage ring is present on both the dorsal and the ventral side. In the vertical transverse section, the two sori are present opposite to each other, and each sorus have many megasporangia in the middle, while 1 to 2 microsporangia at the ends. The mucilage ring is present on the dorsal and the ventral side. If you see in the structure and essence, each micro or megasporangia consists of single layered of jacket enclosing the spore mother cells. At maturity, these mother cells undergoes meiosis and few mitosis to produce the 32 to 64 spores. In microsporangium, all spores survive, while the megasporangium only one spore survive and become very large megaspore. During dehiscence, sporocarps imbibe water from the gelatinous ring swells, expands and pushes out of the split sporocarp. It also pulls the sori, and the sori come out in a row. Indusium gelatinize and liberate the sporangia, and later the sporangial walls also gelatinize and the spores are liberated in the surrounding water. The microspores which are formed in Marselia, these develop into the male gametophyte. The microspores are yellowish, spherical haploid with triradiate ridge and consist of uninucleate cytoplasm surrounded by spore wall. Spore wall is two-layered and endosporium, is endosporium and exosporium. Spore absorbs water and increase considerably in size. Nucleus divide to form a small prothalial cell and a large apical cell. Apical cell divides to form two antheridial initials. Antheridial initials few jacket cells externally and one spermatogenous cells internally. The spermatogenous forms the 16 androcytes which represent the antheridium, and this stays the prothalial cells and the jacket disintegrate, and the two groups of the androcytes remain free within the microspore, and the androcytes develops into multiciliated coiled spermatocyte with vesicles at one end, and development of the male gametophyte here is endosporic. So this is the structure of the male gamete. This is the sori. This is the young Megasporangium towards the with the young Microsporangia. This Microsporangia will divide and they forms the what you call the microspores, which forms the multiflagellated spermatozoids. The female gametophyte is developed from megaspore mother cell. Base Megaspore is elliptical with short papilla at one end. Spore wall is two-layered, endosporium and exosporium. Papilla is surrounded by the endosporium. Nucleus is located in the apical papilla and is surrounded by denser. The rest of the spore is filled with watery cytoplasm and food. Megaspore germinate and give rise to female gametophyte. The development is endosporic type of the phase. So if you see the prothalial cell do not here the transfer the apical nucleus divides into unequal nuclei. One nucleus remains in the dense cytoplasm while the larger one migrates to watery cytoplasm. Transfer wall is formed at the base of the papilla, separating the upper small cell and the lower large cell called the prothalial cell. Now this prothalial cell do not further divide and acts as nutritive cell. The upper cell divide and form the archegonia with short neck and the venter, which has single neck canal cell surrounded by jacket made up of two tiers of the four cells each. Venter contains the venter canal cell which at maturity megaspore absorbs water, and the tip of the megaspore split in triradiate fissure and the archegonium is exposed. Female gametophyte is surrounded by gelatinous mass and a funnel shaped opening at the top. The gametophyte during fertilization, the multiflagellated gametes are liberated from the male gametophyte and are attracted by chemical substances present in the gelatinous mass. Movement of the spermata under the influence of the chemical substances is called chemotaxis. Spermatozoids swarm around and enter the gelatinous matrix of the female gametophyte. Only one spermatozoid enters the open neck and fertilizes the egg to form the diploid zygote. Zygote develops into the embryo. Embryo shows cotyledons and roots develop from upper half of the embryo, while stem and foot develops from the lower half of the embryo. The cotyledons come out of the calyptra. The rhizoids are developed at the first on the root. Very soon the embryo settles in the mud land and mudland, and they develop into a Marselia plant. Here there are, if you see the life cycle, there are two types of the two morphological are shown. One is the what you call the sporophytic plant. The other is known as the gametophytic. So here the type of the life cycle is known as the heteromorphic type of life cycle, or the alternation of generation is known as heteromorphic type. Thank you.